Greetings. This is the final project for BMI 224, Pharmacogenomics. I'm going to talk about the pharmacogenomics of tamoxifen and ESR1. Tamoxifen is primarily used for breast cancer. It's used as a selective estrogen receptor modulator, which means it has different effects in different tissues. It's an estrogen antagonist in breast tissue and an estrogen agonist in other tissues. Used primarily for ER-positive breast cancer, as well as for breast cancer prevention and things as ductal carcinoma in situ. Certainly has saved many lives. First approved for clinical use in 1973, however, a U.S. patent was still issued in 1985 due to a complex legal issue. It has numerous brand names such as Molvidx and Tamoxifen, and dosages in tablets. The main effect of tamoxifen is to inhibit estrogen from binding to estrogen receptors, which inhibits growth of cancer cells. And there are numerous side effects such as hot flashes, higher risk of endometrial cancer, thromboembolic events, and clinical depression. And distribution is 7 to 14 hours, elimination 5 to 7 days. And toxicity has shown some respiratory difficulties and convulsions. The main mechanism of action is rather complex. Tamoxifen itself is a prodrug. It metabolizes in the liver to numerous metabolites that themselves have much greater affinity for estrogen receptors, in fact. Endoxifen is a primary metabolite believed to be responsible for breast cancer treatment success. They all wander to the nucleus and then bind to estrogen receptors for producing a conformational change, which then blocks the expression of estrogen-dependent genes. There is known pharmacogenomic data for CYP2D6 genotypes, but we're going to talk about different genotypes. I'm going to talk about ESR1, which is the primary estrogen receptor. It's a nuclear hormone receptor of a class of over 150 proteins. It has 3 different domains: a modulating N-terminal domain, a DNA binding domain, and a C-terminal ligand binding domain. You see an image of that on the right with estrogen bound. On chromosome 6, 4 different isoforms and 595 amino acids, and you see a red dot on chromosome 6 in the bottom image. In response to tamoxifen, what happens is that tamoxifen changes the activation loop out into an inactive conformation. You can see it on the bottom right, that it's ranging out, thereby stopping the growth signal for estrogen-dependent gene expression. There are 20 characterized SNPs. A few of them are of known pathogenicity, and there's not much population characterization. I'm only going to talk about one SNP in particular, RS9340799, which is an intron. 2 papers looked at this. One did not have a significant p-value, but looked into embolism and thrombosis. The other was looking into cholesterol, triglyceride, and high-density lipoprotein effects, and did have a significant p-value. However, if you look at the summary, there's really not that much evidence. You know, a small sample size, 118 women in the second study I mentioned before, and it's unclear how significant this effect actually is. However, you know, there is potential clinical utility here. Tamoxifen has been used for a long time for breast cancer treatment. It's being used increasingly now as a prophylactic agent, which means that side effects become more important and better predictions of possible outcomes also become more important. However, there are many questions remaining before it can be used clinically, you know, such as, are there other ESR1 SNPs that might be important? More larger patient cohorts and more diverse populations would be needed for the SNP mentioned earlier in order to proceed, and just characterizing just how significant the cholesterol embolism thrombosis effects of these SNPs are. And bibliography, and thank you.